Hi there, my name is Centli Garces, and today I'm going to talk about my poster, which is called Facile Synthesis of Dioxide Titanium and Metal Modified Dioxide Titanium with Photocatalytic and Antibacterial Properties. Well, first of all, I would like to talk about the process based on photocatalysis, which has more and more applications that allow solving worldwide population problems. These applications goes from the production of clean and sustainable energy and elimination of contaminants in water and air, also the feasibility of disinfecting surfaces and environments through inactivation of microorganisms like bacteria and viruses. All of these gaining importance recently due to the health crisis caused by COVID-19. Well, the current work shows results of the synthesis of dioxide titanium doped with different metals. And these metals are iron, nickel, copper, and silver. And they are synthesized by simplified salt gel method in order to improve their optical and antibacterial properties and its effect on the performance of methylene blue photo-degradation and Aureus inhibition test. Well, the general mechanism of photocatalytic process is represented by a diagram in figure number 1. This consists of irradiating the photocatalyst with light. When the photon energy is higher than the excitation energy of the catalyst, it is possible to excite electrons from the valence band, which pass to the conduct band. This causes the existence of positively charged holes in the first of these bands, so that the holes and electrons move to the surface of the photocatalyst and there they react with absorbed species. In such a way, holes can oxidize and reduce species, and electrons can reduce oxidizing species. Well, next I want to talk about the results in figure number 2. First of all, it is known that titanium dioxide can have 3 crystallographic phases, that is, anatase, bruchite, and rutil. As we can see, various results were achieved, all the three of them, by the X-ray diffraction. The most interesting, I think, is the third one, the number C, because this one is belongs to the iron photocatalyst, and as we can see, this one have all the three of the phases, these crystallographic phases. It's really atypical because for the synthesize we use calcination temperatures of 500 Celsius degrees, and it's not normal, or I think it's very atypical that do the calcination temperature at the time of synthesize we obtain bruchite. This phase usually appears at temperatures above 500 Celsius. So, well, going to the figures 3 and 4, we can see the microstructures of titanium dioxide, which present granules. These are characteristic of the catalysis, and on the other hand, we can see how in the figure number 4, the microstructure changed when it was doped with copper and silver, making it more compact. Well, the copper and silver deposits can be seen in this micrograph, as we can see. And in this figure, the number 5, we can see the behavior of the catalyst in moment of degrading methylene blue. The latter is highly reported contaminant that can be degraded by oxidative process. Well, these experimental phases help us to corroborate that our catalysts have the photo-oxidative capacity in the presence of visible light. And why this is important? Well, is that because this entire process is the same by which a microorganism can be degraded. Also, it can be seen the materials with the highest photooxidative capacity are those that were doped with silver and copper, and the catalyst with with nickel. So these materials were selected for the antimicrobial test with Aureus, as we can see in the figure number 6, that it is this one. Well, in this figure, we can see how, after following Kirby-Bauer methodology, Aureus was incubated in enriched agar. Subsequently, 3 filter paper discs were added, which were previously submerged in a solution containing the catalyst to be analyzed. Then one of these Petri box of each catalyst was irradiated with UV light, and the other was left on irradiated, right? And well, it was not radiated. It can be seen that inhibition halos were obtained in all of the samples analyzed, which indicates that all the catalysts have antimicrobial activity to a greater or less extent. Well, materials with silver deposit stands out, as well as those that were irradiated with UV light. It should be noted that copper has antibacterial properties by itself, so it's kind of expected that has these inhibition halos. We consider that for the catalyst with copper and silver deposit, the inhibition is carried out by 2 different mechanisms. The first mechanism is a mechanism of inhibition by photocatalysis, and the second one is a mechanism of rupture of the cell membrane by action of the silver, since that the cell membrane has a value in millivolts, which is negative, and since the silver nanoparticles has a positive charge, they adhere to the cell membrane of the bacteria. The nanoparticles create an imbalance in the cell membrane, which causes a hole through which the nanoparticles enter, causing an attack of DNA reproduction. This effect is known as death by lysis. In addition to the above, the irradiation of UV light with a wavelength below 385 nanometers activates photocatalysis by the photons of this light, which promotes that the holes generated by the cell membrane are held. So, as conclusions here, we have in the photocatalytic evaluation of our catalysis with nickel was the most active material, reaching 20% of methylene blue degradation after 3 hours of visible light irradiation, followed by the catalysis with copper and silver, which reached 12% of degradation. The results of inhibition of Aureus, like I said, show that all the tested materials present inhibition halos, but the best results were obtained with the one who has copper and silver, showing an extra increasing of the halo after UV light irradiation treatment, demonstrating that the additional photocatalytic effect in the inactivation of bacteria. Well, that's all. This is my reference, and if you want to contact me or reach me and tell me whatever comments or even questions, you can reach me here. This is my mail. So that's it. Thanks so much for listening to me, and see you another time.